The human brain, a universe within, a labyrinth of cells, a symphony of electrical impulses. It is the command center of our being, the seat of our consciousness, the wellspring of our thoughts, emotions, and actions. For centuries, this enigmatic organ has captivated philosophers, scientists, and thinkers alike, its intricate workings shrouded in mystery. What is it about this 3-pound mass of tissue that allows us to perceive the world, to feel the warmth of the sun, to compose symphonies that stir the soul? How does the brain, a biological machine seemingly no different from any other organ, give rise to the subjective experience of being alive, of having a self? It is a question that has echoed through the ages, a question that continues to fuel our insatiable curiosity. The brain, with its billions of neurons firing in concert, orchestrates the complex tapestry of our lives. From the simplest of reflexes to the loftiest of aspirations, every aspect of our existence is mediated by this remarkable organ. Yet for all its power, the brain remains an enigma, a puzzle box with secrets yet to be unlocked. But the journey of discovery is well underway. Scientists around the world are working tirelessly, using increasingly sophisticated tools and techniques to unravel the mysteries of the brain. And with each new discovery, we gain a deeper appreciation for the wonders of this extraordinary organ. Imagine for a moment the experience of a newborn baby. The world, once a muted symphony of sensations within the womb, explodes into a cacophony of light, sound, and touch. The brain, a blank slate at birth, begins its lifelong task of processing this sensory information, making sense of the chaos, and constructing a coherent picture of reality. It is a process of constant learning, adaptation, and refinement, a testament to the brain's remarkable plasticity. From the moment we take our first breath, our brains are bombarded with an overwhelming torrent of data. Our senses, like tireless reporters on the front lines, relay a constant stream of information about the external world. The smell of freshly baked bread, the gentle caress of a summer breeze, the vibrant colors of a sunset, all are faithfully transmitted to the brain, where they are decoded, interpreted, and woven into the fabric of our conscious experience. But the brain's role extends far beyond merely processing sensory input. It is also the seat of our emotions, the crucible in which our feelings are forged. Joy, sadness, anger, fear, these emotions, so fundamental to the human experience, are all orchestrated by the brain. They color our perceptions, shape our decisions, and drive our actions. And then there is the realm of thought, the ability to form ideas, to reason, to dream, to imagine worlds beyond our own. This uniquely human capacity, the ability to engage in abstract thought, is perhaps the most profound mystery of the brain. How does a network of interconnected cells give rise to the spark of consciousness, to the ability to ponder our own existence, to contemplate the vastness of the universe? The study of the brain is, in many ways, the ultimate frontier of scientific exploration. It is a journey into the very essence of what it means to be human, a quest to understand the biological underpinnings of our thoughts, emotions, and behaviors. And while we have made remarkable strides in recent decades, the brain, with its staggering complexity, continues to challenge our assumptions and push the boundaries of our understanding. This exploration is not merely an academic pursuit, it has profound implications for our health and well-being. By understanding how the brain works, we can develop new treatments for neurological and psychiatric disorders that afflict millions worldwide. We can gain insights into the aging process, unlock the secrets of memory, and perhaps even enhance our cognitive abilities. The journey ahead will undoubtedly be challenging. The brain is, after all, the most complex object in the known universe. But the potential rewards are immeasurable. By unraveling the threads of the brain's intricate tapestry, we stand to gain not only a deeper understanding of ourselves, but also the tools to alleviate suffering and unlock the full potential of the human mind. Imagine the human brain as a walnut, its wrinkled surface a testament to its intricate complexity. This outermost layer, the cerebral cortex, is the seat of our most sophisticated cognitive abilities. It is here, in this thin sheet of neural tissue, that we think, plan, reason, and dream. The cerebral cortex, with its characteristic folds and grooves, is divided into two hemispheres, left and right, each responsible for controlling the opposite side of the body. These hemispheres, though seemingly mirror images of each other, are not identical in their functions. The left hemisphere, in most individuals, is dominant for language, logic, and analytical thinking, while the right hemisphere excels in spatial awareness, creativity, and emotional intelligence. Yet these hemispheres do not operate in isolation. They are in constant communication, exchanging information through a thick band of nerve fibers called the corpus callosum, ensuring that both sides of the brain work in harmony. The cerebral cortex is further divided into four lobes, each specialized for different functions: the frontal lobes, the parietal lobes, the temporal lobes, and the occipital lobes. These lobes, like specialized departments within a vast organization, work together seamlessly, their interactions giving rise to the rich tapestry of human thought and behavior. The frontal lobes, located at the front of the brain, are the seat of our executive functions, the command center for planning, decision-making, and voluntary movement. Behind the frontal lobes lie the parietal lobes, responsible for processing sensory information from the body, such as touch, temperature, and pain. The temporal lobes, located on the sides of the brain, play a crucial role in memory, language comprehension, and auditory processing. And finally, at the back of the brain reside the occipital lobes, dedicated to processing visual information from the eyes. Together, these four lobes form the intricate neural network that allows us to perceive the world, interact with our environment, and navigate the complexities of human experience. Section 2. The frontal lobes, orchestrating our actions and decisions. The frontal lobes are the brain's executive control center, responsible for planning, decision-making, and impulse control. Damage to the frontal lobes can profoundly affect personality and behavior. They guide and coordinate brain activities like a conductor leading an orchestra. Frontal lobe damage may cause impulsivity, poor judgment, and emotional instability. They help us learn from mistakes and make sound judgments. Section 3. The parietal lobes, mapping our sensory world. Behind the frontal lobes lie the parietal lobes. They integrate sensory information, allowing us to experience touch, temperature, pain, and pressure. Imagine them as the brain's sensory map makers. Close your eyes and feel the texture of your clothing. All these sensations are processed by the parietal lobes. They actively interpret and integrate sensory information. For instance, when you reach for a cup of coffee. Damage to the parietal lobes can result in impairments. One such disorder is hemispatial neglect. People with this condition may eat from only one side of their plate. The parietal lobes also play a crucial role in proprioception. This sense allows us to walk, run, and dance. Without them, our movements would be clumsy and uncoordinated. Section 4. The temporal lobes, guardians of memory and language. Nestled beneath the lateral fissures lie the temporal lobes. They process auditory information, comprehend language, and form new memories. They are the keepers of our past experiences and interpreters of words. Imagine the temporal lobes as vast libraries, storing a lifetime of memories. These memories shape our identities and guide our decisions. Within the temporal lobes lies the hippocampus, crucial for forming new memories. The hippocampus binds sensory information into coherent episodes. Damage to the hippocampus can result in profound memory impairments. Individuals with anterograde amnesia may be unable to form new memories. The temporal lobes are also home to Wernicke's area, crucial for language comprehension. Damage to Wernicke's area can result in Wernicke's aphasia, affecting language understanding. People with Wernicke's aphasia may produce nonsensical speech. The temporal lobes are essential for communication, learning, and memory. At the back of our heads, nestled beneath the parietal lobes, lie the occipital lobes. These paired structures are the primary visual processing centers of the brain, responsible for receiving, interpreting, and translating the raw data from our eyes. into the meaningful visual world we perceive. Imagine the occipital lobes as sophisticated image processors, converting the two-dimensional patterns of light that fall on our retinas into a vibrant three-dimensional representation of our surroundings. The journey of visual information from the eyes to the occipital lobes is a remarkable feat of neural engineering. Light entering our eyes is first focused by the cornea and lens onto the retina, a light-sensitive layer of tissue lining the back of the eye. Here, specialized cells called photoreceptors convert light energy into electrical signals, which are then transmitted through a complex network of nerve fibers to the optic nerve. The optic nerves from each eye meet at a point called the optic chiasm, where some of the nerve fibers cross over to the opposite side of the brain. This crossing over ensures that the left visual field from both eyes is processed by the right occipital lobe, and the right visual field from both eyes is processed by the left occipital lobe. Once the visual information reaches the occipital lobes, it is analyzed and interpreted in a hierarchical manner. Simple features such as lines, edges, and orientations are detected first, followed by more complex features such as shapes, objects, and faces. This information is then integrated with input from other brain regions, such as those involved in memory and attention, allowing us to recognize familiar objects, identify faces, and navigate our environment. Damage to the occipital lobes can result in a variety of visual impairments, ranging from partial blindness in specific areas of the visual field to complete blindness, depending on the location and extent of the damage. Tucked away beneath the occipital lobes at the back of the brain lies the cerebellum, a structure often overlooked but crucial for our ability to move with grace, coordination, and precision. Though smaller than the cerebrum, the cerebellum is densely packed with neurons, containing nearly half of all the neurons in the brain. Imagine the cerebellum as a skilled dancer, effortlessly coordinating the movements of countless muscles, ensuring that our actions are smooth, fluid, and perfectly timed. Unlike the cerebral cortex, which initiates voluntary movements, the cerebellum does not directly command our muscles. Instead, it receives information from the sensory systems, the spinal cord, and the cerebral cortex about the body's position, intended movements, and the environment. The cerebellum then uses this information to fine-tune motor commands, ensuring that movements are executed smoothly, accurately, and with appropriate timing. Think of the cerebellum as the brain's error correction system for movement. It constantly compares our intended movements with our actual movements, making subtle adjustments to ensure that our actions match our intentions. This continuous feedback loop allows us to learn new motor skills, adapt to changing environments, and maintain balance and posture without conscious effort. The cerebellum's role in motor control becomes strikingly evident when it is damaged. Individuals with cerebellar damage often exhibit ataxia, a condition characterized by uncoordinated, jerky movements, difficulty maintaining balance, and tremors. Simple tasks such as walking, reaching for objects, or speaking can become challenging, requiring conscious effort and concentration. Interestingly, the cerebellum's influence extends beyond motor control. Recent research suggests that the cerebellum may also play a role in cognitive functions such as language, attention, and emotional regulation. For instance, studies have shown that individuals with cerebellar damage may exhibit difficulties with language processing, particularly with timing and rhythm, suggesting that the cerebellum's role in timing extends beyond motor control to encompass cognitive functions as well. Deep within the brain, nestled beneath the cerebral cortex, lies the limbic system, a collection of interconnected structures that plays a central role in our emotional lives, shaping our feelings, motivations, and memories. Imagine the limbic system as the brain's emotional command center, orchestrating a complex symphony of physiological and behavioral responses to internal and external stimuli. The limbic system is not a single unified structure, but rather a network of interconnected brain regions, each with its own specialized functions. These regions include the amygdala, the hippocampus, the thalamus, the hypothalamus, and the cingulate gyrus. Together, these structures work in concert to process emotional information, regulate our responses to stress and reward, and form long-lasting memories of emotionally significant events. The amygdala, a small almond-shaped structure located deep within the temporal lobes, plays a crucial role in processing fear and other emotions. It acts as a rapid threat detection system, receiving sensory information from the thalamus, a relay station for sensory input, and the sensory cortex, which processes detailed information about the environment. If the amygdala detects a potential threat, it can trigger a cascade of physiological and behavioral responses, such as increased heart rate, rapid breathing, and the release of stress hormones like adrenaline and cortisol, preparing the body for a fight or flight response. The hippocampus, a seahorse-shaped structure located in the temporal lobes, plays a critical role in forming new memories, particularly those associated with emotional experiences. It receives input from the amygdala and other sensory areas of the brain, linking emotional significance to events and experiences. This connection between the amygdala and the hippocampus helps to explain why we tend to remember emotionally charged events more vividly than mundane ones. The thalamus, a pair of oval-shaped structures located in the center of the brain, acts as a relay station for sensory information, directing incoming sensory signals to the appropriate areas of the cortex for processing. Deep within the temporal lobes lies the amygdala, a small but mighty structure crucial for our emotional lives, especially fear and anxiety. Imagine the amygdala as the brain's vigilant watchdog, constantly scanning for threats. Its role in fear processing is deeply rooted in our evolutionary history. For our ancestors, detecting and responding to threats was essential for survival. A rustle in the bushes could signal a predator. Quick reactions were crucial for survival. Today, the amygdala still keeps us safe, even with less immediate threats. A speeding car or barking dog can trigger the amygdala's alarm system. It also helps us learn to fear new things. Fear conditioning occurs when a neutral stimulus becomes associated with fear. For example, a dog bite can lead to a lifelong fear of dogs. The amygdala strengthens connections between neurons representing fear and the stimulus. The amygdala's influence extends beyond fear processing. It also plays a role in other emotions and social cognition. The amygdala helps us navigate the social world by processing emotional information. Curled like a seahorse deep within the temporal lobes lies the hippocampus, a small but mighty structure that plays a pivotal role in our ability to form new memories, learn from our experiences, and navigate the world around us. The hippocampus is the brain's master architect, constructing mental maps of our experiences and weaving them into memory. Essential for episodic memory, it helps us recall specific events and experiences. These memories are rich, multifaceted, and vivid. Think back to a favorite childhood memory. The hippocampus allows us to relive those moments vividly. It also plays a crucial role in spatial navigation. Imagine navigating a new city without a map. You might wander aimlessly, struggling to remember landmarks. The hippocampus acts like our internal GPS. It helps us navigate familiar environments with ease. London taxi drivers have larger hippocampi, showing spatial navigation can change the brain. The hippocampus works with other brain regions to encode, store, and retrieve memories. The amygdala adds emotional significance to memories. The prefrontal cortex helps organize and retrieve memories. The sensory cortices provide the raw materials of memory. Deep within the brain lies the hypothalamus, a tiny but powerful structure maintaining the body's internal balance. Imagine the hypothalamus as the body's master conductor, orchestrating hormones and neurochemicals. It connects to the pituitary gland, influencing the endocrine system and autonomic functions. The hypothalamus translates neural signals into hormonal commands, affecting organs and tissues. It regulates body temperature, acting like an internal thermostat. Monitoring core temperature, it initiates responses to maintain stability. When hot, it dilates blood vessels and triggers sweating to cool the body. Sweating cools the body through evaporation. In cold, it constricts blood vessels and triggers shivering to generate heat. The hypothalamus regulates hunger and satiety. It receives signals about nutrient levels and energy stores. Releasing hormones, it stimulates or suppresses appetite. This ensures we meet energy needs without overeating. The hypothalamus also regulates the sleep-wake cycle. Imagine the human brain, a universe within, a labyrinth of cells, a symphony of electrical impulses. Neurons, the brain's building blocks, transmit information, enabling us to think, feel, and interact. Each neuron has a cell body, dendrites, and an axon, forming a vast communication network. Myelin-covered axons speed up signal transmission, crucial for efficient communication. Neurons communicate at synapses, converting electrical signals to chemical ones. This intricate dance forms the basis for all our thoughts, feelings, and actions. Imagine a bustling city, its streets teeming with cars, buses, and pedestrians, all navigating a complex network of intersections and traffic signals. This intricate dance of movement and communication finds its parallel in the brain, where billions of neurons communicate with each other at specialized junctions called synapses. These tiny gaps between neurons are where the language of the brain, a complex interplay of electrical and chemical signals, is spoken. When an action potential, the electrical impulse that travels down a neuron's axon, reaches the axon terminal, it triggers the release of neurotransmitters, chemical messengers that carry signals across the synapse. These neurotransmitters, stored in small sacs called vesicles within the axon terminal, are released into the synaptic cleft, the tiny gap between the axon terminal of the sending neuron and the dendrite of the receiving neuron. Once released into the synaptic cleft, neurotransmitters diffuse across the gap and bind to specialized receptors on the dendrite of the receiving neuron. These receptors, like locks that can only be opened by specific keys, are designed to bind to particular neurotransmitters, ensuring that the right signals are transmitted between neurons. The binding of a neurotransmitter to its receptor can have either an excitatory or an inhibitory effect on the receiving neuron. Excitatory neurotransmitters, like glutamate, increase the likelihood that the receiving neuron will fire an action potential, passing the signal along. Inhibitory neurotransmitters, like GABA, have the opposite effect, decreasing the likelihood that the receiving neuron will fire, thus dampening neural activity. The balance between excitatory and inhibitory signals in the brain is crucial for proper brain function. Too much excitation can lead to seizures, while too much inhibition can result in lethargy and coma. The brain constantly adjusts the strength of synaptic connections, strengthening some and weakening others, in response to experience, a process known as synaptic plasticity. Imagine a vast orchestra, its musicians playing a symphony of sound, each instrument contributing its unique voice to the harmonious whole. In the brain, a similar symphony unfolds, orchestrated not by instruments but by neurotransmitters, the brain's chemical messengers, each with its own distinct role in shaping our thoughts, feelings, and actions. Neurotransmitters released from the axon terminals of neurons carry signals across synapses, the tiny gaps between neurons, influencing the activity of neighboring neurons, muscles, or glands. These chemical messengers are the brain's way of communicating, their diverse effects shaping our moods, regulating our movements, and influencing our perceptions. One of the most abundant neurotransmitters in the brain is glutamate, the primary excitatory neurotransmitter responsible for stimulating neural activity. Glutamate plays a crucial role in learning and memory, and its overactivity is implicated in neurological disorders such as epilepsy. GABA, on the other hand, is the brain's primary inhibitory neurotransmitter, acting as a brake on neural activity, preventing overexcitation. GABA promotes relaxation and reduces anxiety, and its deficiency is linked to anxiety disorders and epilepsy. Dopamine, often referred to as the feel-good neurotransmitter, is involved in reward, motivation, and movement. Dopamine is released in response to pleasurable experiences, reinforcing behaviors that lead to reward. Parkinson's disease, a neurological disorder characterized by tremors and difficulty initiating movements, is caused by the death of dopamine-producing neurons. Serotonin plays a crucial role in mood regulation, sleep, appetite, and aggression. Low levels of serotonin are implicated in depression, while antidepressants such as selective serotonin reuptake inhibitors, SSRIs, work by increasing serotonin levels in the brain. Acetylcholine, the first neurotransmitter to be discovered, is involved in muscle movement, learning, memory, and attention. Alzheimer's disease, a neurodegenerative disorder characterized by memory loss and cognitive decline, is associated with a decrease in acetylcholine levels in the brain. Imagine a spark traveling down a fuse, igniting a chain reaction that culminates in a burst of light and sound. This explosive release of energy finds its parallel in the brain, where neurons communicate through a remarkable electrochemical process known as the action potential. This rapid and transient change in electrical potential across a neuron's membrane is the fundamental unit of communication in the nervous system, the spark that ignites our thoughts, feelings, and actions. The action potential is an all-or-nothing event, like flipping a light switch on or off. It is triggered when the electrical potential difference across the neuron's membrane, known as the membrane potential, reaches a certain threshold, typically around -55 millivolts. This threshold is reached when the neuron receives enough excitatory input from other neurons. When the threshold is reached, voltage-gated sodium channels, protein channels embedded in the neuron's membrane that are sensitive to changes in voltage, open rapidly, allowing an influx of positively charged sodium ions into the neuron. This influx of positive charge causes the membrane potential to depolarize rapidly, becoming more positive. As the membrane potential becomes more positive, voltage-gated potassium channels, another type of ion channel, open more slowly. Potassium ions, also positively charged, flow out of the neuron, driven by both the concentration gradient and the electrical gradient. This outflow of positive charge causes the membrane potential to repolarize, returning to its resting state. The action potential doesn't stop there. The potassium channels often remain open for a brief period after the membrane potential has returned to its resting state, causing the membrane potential to become even more negative than usual, a process known as hyperpolarization. During this refractory period, it is difficult or impossible for the neuron to fire another action potential. Imagine a sculptor molding clay, shaping and reshaping it in response to their creative vision. The brain, like clay in the hands of a sculptor, possesses a remarkable ability to change its structure and function in response to experience, a phenomenon known as neural plasticity. This adaptability allows us to learn new skills, recover from injuries, and adapt to changing environments. Neural plasticity involves mechanisms that modify connections and rewire circuits. These mechanisms operate at multiple levels, from synapse changes to new neuron formation. Imagine a sudden threat, a speeding car veering towards you, or a snarling dog blocking your path, a looming deadline triggering anxiety. The sympathetic nervous system activates, preparing us for action. It sends signals through our nerves, readying our bodies. Our hearts race, pumping blood to our muscles. Breathing quickens, fueling our movements. Pupils dilate, enhancing vision. Blood sugar rises, providing energy. Digestion slows, diverting energy to vital functions. Blood flow redirects to muscles and brain. Muscles tense, poised for action. Palms sweat, increasing grip. This response, honed by evolution, is essential for survival. It also plays a role in excitement and joy. Section 2. The parasympathetic nervous system, rest and digest. Imagine yourself in a state of deep relaxation. Your heart beats slowly and steadily. Your breathing is deep and rhythmic. Your muscles are loose and relaxed. Your mind is at ease. This state of tranquility is the domain of the parasympathetic nervous system. It promotes rest, repair, and digestion. The parasympathetic nervous system works with the sympathetic nervous system to maintain balance. If the sympathetic nervous system is the accelerator, the parasympathetic nervous system is the brake. It allows us to unwind and replenish our energy. Promoting long-term health and well-being, the parasympathetic nervous system enhances digestion. It promotes relaxation and reduces anxiety. Like the sympathetic nervous system, the parasympathetic nervous system influences various organs and glands. Its effects are generally the opposite of the sympathetic nervous system. Where the sympathetic nervous system speeds up the heart rate, the parasympathetic nervous system slows it down. Where the sympathetic nervous system quickens breathing, the parasympathetic nervous system slows it down and deepens it. Where the sympathetic nervous system diverts blood flow away from digestion, the parasympathetic nervous system enhances it, promoting the breakdown and absorption of nutrients. The parasympathetic nervous system promotes relaxation, reduces anxiety, and conserves energy. It allows us to fall asleep at night, to recover from illness or injury, and to experience a sense of calm and well-being. The sympathetic and parasympathetic nervous systems are constantly engaged in a delicate dance. Their opposing actions working together to maintain homeostasis, the body's internal balance. When we encounter a threat, the sympathetic nervous system takes the lead, preparing us to fight or flee. Once the threat has passed, the parasympathetic nervous system steps in, calming our bodies and restoring equilibrium. Section 3. The Symphony of Speech. From thought to articulation. Imagine the effortless flow of conversation, thoughts cascading into words, ideas into narratives. Speech is a complex symphony of neural activity, transforming thoughts into spoken words. It begins in the brain's language centers, primarily in the left hemisphere. Broca's and Wernicke's areas formulate thoughts into sentences and retrieve words. The signal travels to the motor cortex, controlling voluntary movements. The motor cortex maps our body, controlling speech muscles. The signal travels through the brainstem and spinal cord. The cerebellum and basal ganglia refine and modulate the signal. Finally, the signal reaches the speech muscles, producing sound. Lips, tongue, jaw, and vocal cords work together, creating speech. This intricate process unfolds in milliseconds, showcasing the brain's coordination. Speech allows us to share ideas, express emotions, and connect with others. Section 4. Broca's area, the conductor of language production. Imagine a conductor guiding an orchestra. Broca's area in the brain orchestrates our speech, transforming thoughts into words. Named after Pierre-Paul Broca, this area is crucial for language production and grammatical processing. Damage to Broca's area results in Broca's aphasia, causing difficulty in speaking fluently. People with Broca's aphasia understand language but struggle to express themselves. Their speech is often halting and lacks natural rhythm. This can be frustrating and isolating. Broca's area also plans and sequences grammatical structures. It ensures our words are arranged meaningfully. This is crucial for conveying complex ideas. Broca's area works with other language regions like Wernicke's area for comprehension. Section 5. Wernicke's area deciphering the code of language. Imagine listening to a foreign language. Its sounds washing over you. The words a jumble of unfamiliar syllables. Wernicke's area in the brain helps us understand spoken and written language. Damage to this area can cause Wernicke's aphasia, where speech is fluent but nonsensical. Located in the temporal lobe, it plays a crucial role in language comprehension. Named after Carl Wernicke, this area is essential for understanding language's nuances, like sarcasm and metaphors. The intricacy of fine motor movements. Writing your name involves precise muscle control. Fingers glide, muscles contract and relax. Fine motor skills showcase brain's control. Practice strengthens neural pathways. Skills improve with repetition. Fine motor skills enrich our lives. Section 2. The cerebellum's role in precision and timing. Imagine a tightrope walker gracefully traversing a thin wire high above the ground, each step perfectly balanced, each movement fluid and controlled. This remarkable feat of balance, coordination, and timing would be impossible without the cerebellum, a small but mighty structure located at the back of the brain, often referred to as the little brain due to its resemblance to the cerebrum. The cerebellum, despite its diminutive size, contains more than half of the brain's neurons, a testament to its crucial role in motor control. Unlike the cerebral cortex, which initiates voluntary movements, the cerebellum does not directly command our muscles. Instead, it acts as a behind-the-scenes coordinator, fine-tuning our movements, ensuring that they are smooth, accurate, and perfectly timed. As we perform a movement, whether it's reaching for a cup of coffee or executing a complex dance routine, the cerebellum receives a constant stream of information from our senses, our muscles, and the motor cortex, the brain region responsible for planning and initiating movements. It compares this incoming information with the intended movement, detecting any discrepancies and sending corrective signals back to the motor cortex, ensuring that our actions match our intentions. The cerebellum's role in timing is particularly crucial for tasks that require precise coordination, such as playing a musical instrument, typing on a keyboard, or hitting a tennis ball. It acts like an internal metronome, providing the precise timing cues needed to execute these movements with speed and accuracy. Damage to the cerebellum can result in ataxia, a condition characterized by uncoordinated, jerky movements, difficulty maintaining balance, and tremors. Deep within the brain, the basal ganglia regulate movement, motivation, and reward. Imagine the basal ganglia as skilled mechanics fine-tuning our movements. They initiate and smooth movements, selecting appropriate actions. They receive input from the cerebral cortex and substantia nigra, processing goals and intentions. The basal ganglia send signals back, influencing movement. They regulate muscle tone, maintaining posture and preparing for movement. Disruptions can lead to disorders like Parkinson's and Huntington's. They help in learning new motor skills and forming habits. The basal ganglia are also involved in motivation, reward, and addiction. Imagine the human brain, a universe within a labyrinth of cells, a symphony of electrical impulses. It is the command center of our being, the seat of our consciousness, the wellspring of our thoughts, emotions, and actions. Every aspect of our behavior is orchestrated by this remarkable organ, shaping who we are and how we interact with the world. Our experiences shape the structure and function of our brains, a phenomenon known as neuroplasticity. This adaptability allows us to learn, adapt, and develop new skills. Our emotions, from joy to fear, are complex responses preparing us to act. Fear triggers physiological changes, preparing us to fight or flee. The brain enables us to navigate human relationships and form bonds. Brain injury can result in behavioral changes, highlighting the brain's role. The brain's influence on behavior is a testament to its extraordinary capabilities. As we unravel the brain's mysteries, we appreciate the interplay of factors shaping us. As we conclude our journey into the depths of the human brain, we find ourselves standing at the edge of a vast and uncharted territory, humbled by the sheer complexity and elegance of this extraordinary organ. We have explored the brain's intricate anatomy, delved into the symphony of electrical and chemical signals that animate our thoughts and actions, and marveled at its remarkable capacity for change and adaptation. Yet, for all that we have learned, the brain continues to hold countless mysteries, its secrets beckoning us to venture further, to push the boundaries of our understanding and to unlock the full potential of the human mind. The more we learn about the brain, the more we realize how much more there is to discover. The brain is not a static, unchanging organ, but rather a dynamic, ever-evolving landscape shaped by our genes, our experiences, and the constant interplay between nature and nurture. Every thought we think, every memory we form, every skill we learn leaves its imprint on the brain, altering its structure and function in subtle but profound ways. Despite the remarkable progress that has been made in neuroscience, many fundamental questions remain unanswered. How does the brain give rise to consciousness, that subjective experience of being, of having a self? What are the neural mechanisms underlying our emotions, our motivations, our sense of self? How do our brains allow us to navigate the complexities of human relationships, to form bonds of love and attachment, to experience empathy and compassion? These are not merely academic questions, they are at the heart of what it means to be human. By unraveling the mysteries of the brain, we stand to gain not only a deeper understanding of ourselves, but also the tools to alleviate suffering, to enhance human potential, and to create a more just and compassionate world. The future of brain exploration is bright with promise, fueled by technological advances. New imaging techniques allow us to observe the brain in action. Optogenetics provides unprecedented control over brain circuits. AI helps analyze vast data sets to identify brain activity patterns. These advances open new possibilities for treating brain disorders. The journey into the human brain is just beginning.